In this short feature, I will be describing to you a critical figure in the article by Sato and colleagues, in which a mutant variant of HRD1P was shown to be deficient for the degradation of an integral membrane protein called HMG2P. In the following slides, green will represent any analysis of a gel. Purple will similarly indicate information that is located on graphs, and conclusions will be highlighted for you in yellow. Lastly, any critical results for the interpretation of a figure panel will be indicated in red. First off, in panel A, the authors performed a cyclohexamide chase and measured the amount of an integral membrane protein called HMG2P, which, as I previously discussed, follows the ERAD-M pathway. You can recall from our first test how a cyclohexamide chase works. In short, by adding cyclohexamide to a culture, protein expression is inhibited. From there, you can measure the amount of protein in the cell after fixed time intervals. This is a useful way for displaying the efficiency of protein degradation by ERAD. To account for the differential expression levels in mutant transfectants, the steady-state protein level was measured relative to the wild-type expression. Looking closely at the gel, you can see beneath the wild-type heading the gradual decrease in intensity of the band from 0 to 120 minutes. This is an indication that the HMG2P protein is gradually being degraded by ERAD. Moving to the right, the next heading represents a mutant form of HRD1P in which serine 97 to serine 98 have both been mutated to alanine. These mutations appear to have lessened the degradation, as you can see a dark band even after 120 minutes has passed. This pattern was also seen for another mutant in which aspartic acid 199 was mutated to alanine. The final heading, 3A, on the far right, is a combination of these three mutations in an HRD1P variant that resulted in no apparent change in protein levels after 120 minutes, appearing as a consistent dark band in the three last lanes on the gel. However, you might be thinking, what's black is black, right? Well, since HMG2P was tagged with a green fluorescent protein, the authors could quantify the amount of protein present in each by measuring fluorescence. The results can be visualized in the graph below, where you can see the rate of change of the amount of HMG2P protein remaining over time. As you can see, there is a strong negative slope for the wild-type HRD1P protein. In contrast, each of the mutants have a slope that differed from the wild-type, and when combined in the 3A mutant, indicated in the red circle, approximately 80% of the protein was still left over after 120 minutes. In short, panel B shows the same experiment, except this time the 3A mutant is compared to a C399S HRD1P mutant, which acts as a positive control. This is because it has previously been shown by Bayes and colleagues in 2001 that the C399S HRD1P mutant lacks the ability to ubiquitinolate all ERAD substrates, and thus they are not targeted to the proteasome for degradation. Shown in red on the graph, you can see that the 3A mutant closely resembles the slope of the positive control and not that of the wild type. Again, in panel 3C, you can see a similar experiment has been performed. Interestingly here, though, they used a terminally misfolded version of HMG2P called 6-NYC HMG2P GFP. As you can see from the results of the graph, the wild-type slope has become even more negative in comparison to when the protein was properly folded. This can be expected because with the introduction of the misfold in the protein, there is increased signaling for ERAD degradation. Also, you can notice that the phenotype of the 3A mutant is consistent and resembles that of the positive control. So now that they knew their 3A HRD1P mutant was deficient in ERAD, Sato and colleagues next wanted to investigate which step in the pathway is affected by this mutation. To do this, they performed a ubiquitinylation assay. Remember that by adding a ubiquitin oligomer, the misfolded protein is targeted to the proteasome for degradation. So in this gel, they immunoblotted for the presence of this ubiquitin group. They tested ubiquitinylation under normal conditions and also in the presence of zargosic acid, which was previously shown by Hampton and Bacta in 1997 to increase the signaling for HMG2P degradation. So under the wild-type heading, you can see the normal ubiquitinylation smear and the expected increase in intensity of that smear when the zargosic acid was added. This is indication of that ubiquitinylation did occur. For the C399S mutant, however, there was no ubiquitinylation. This is also expected since the HRD1P variant, C399S, was previously shown to be non-functional for ubiquitinylation. Finally, the most important part of this figure is seen in red. You can see that there was no ubiquitinylation from the 3A HRD1P mutant. So looking back at the ERAD-M pathway, you can see that there is an effect of this mutation at or before ubiquitinylation occurred. There are three possible explanations for the results. The 3A mutation could have caused the HRD1P protein to become non-functional, similar to the C399S mutant, which was unable to ubiquitinylate ERAD substrates. Also, this 3A mutation could have caused the gene to become hypomorphic or less expressed than the wild-type protein, and thus would result in the decrease in ERAD efficiency. Lastly, the 3A residues of the mutant HRD1P protein could have been involved in the recognition of the HMG2P protein. In panel E, the authors tested the second explanation by transforming cells that lacked the HRD1P ubiquitin ligase with HRD1P variants behind a high-expression promoter called TDH3. They then exposed the cells to cyclohexamide and followed up by measuring protein degradation as was previously described. The results show that wild-type degradation of HMG2P-GFP was the same behind the TDH3 promoter as it was behind the native promoter. The same thing occurred with the C399S mutant, as impaired degradation could still be seen, as well as with the 3A mutant. You can see it boxed in red. There is little change in the band intensity from 0 to 2, indicating that the degradation of the HMG2P protein was still impaired even when the protein was expressed 20-fold. This means that the gene is not hypermorphic, and so we can cross this possibility for an explanation off of our list. So what have we learned from Figure 3? In panel A, they identified a 3A mutant HRD1P protein that was deficient in the ERAD degradation of HMG2P membrane protein. This was measured against an ERAD deficient positive control in B, and in panel C, the mutant's effect was shown to be even stronger when the signal for ERAD degradation was increased using a misfolded version of HMG2P. In panel D, they found out that the 3A mutation affected ERAD of the HMG2P protein, before or at the ubiquitinylation step in the ERAD-M pathway. And lastly, in panel E, we found out that this is not a hypomorphic mutation.